Okay, good morning. I'm back. All right, so on Monday, you heard about traffic into the lysosome of the LDL particle and how that ultimately controls cholesterol biosynthesis. That lecture is a little bit misplaced, but we will return to that topic next week when I discuss the biosynthesis of proteins through the secretory pathway that enter into the lysosome. But I want to take a step back to finish a topic that I started a few days ago, and that is how the signal hypothesis can be used to explain the insertion of the three different categories of membrane proteins that I listed in my last lecture. The type 1 protein, which has an N-terminal domain that faces the outside of the cell, and a cytoplasmic domain that faces the inside of the cell, or as that protein is made, the N-terminus sticks into the lumen of the ER, and the C-terminus still faces the cytoplasm. That's type 1. Type 2 is the flip side of that. And type 3 is a protein that snakes its way through the membrane. So I began to describe this with the running through the pathway as it applies, the translocation pathway, as it applies to a secretory protein, the N-terminal domain, the N-terminal signal peptide attached to a otherwise completely soluble protein. Now I'm going to tell you about the type 1 protein, and we'll return to the beginning and discuss again the signal peptide. So here is a simple cartoon diagram of the translocation pore. The N-terminal signal peptide, again, embeds into the channel as a loop, and the hydrophobic residues begin to protrude. Oops, wrong color. And being hydrophobic, the signal peptide tends to diffuse out of the channel. The channel is kind of like a C-shaped structure, where the inside is the polar domain, and the opening between two subunits of the channel is neighboring the phospholipid bilayer. So a signal peptide that inserts initially into the channel, being hydrophobic, will partition out of the channel through the gap between the two subunits into the adjacent bilayer. But the signal peptide, just like my finger and thumb, will still be attached to the N-terminus of the secretory protein. So that will remain in the channel even after the enzyme signal peptidase cleaves at the junction between the signal peptide and the mature protein. So after that, the signal peptide floats away through the channel into the adjacent bilayer, where it's degraded to amino acids. Now it's gone. And the mature N-terminus now begins its course into the lumen of the ER. Now, for a type 1 protein, recall, with the N-terminus facing the outside of the cell, or initially the lumen of the ER, there must be at least one membrane anchor domain that's going to lodge that protein in the bilayer. And so we can imagine that N-terminal domain, that the hydrophobic domain, the 20 amino acid stretch that comprises an alpha helix that's going to keep this in the membrane, may be synthesized by the ribosome, and it may also now, eventually, find its way into the channel. But in this case, a significant portion of the N-terminus has already translocated into the lumen, and now you've got a situation where an apolar stretch of amino acids is stuck in a hydrophilic channel, and just like the signal peptide, that apolar stretch will partition out of the channel into the adjoining bilayer, but in so doing, it will carry with it the polar domain that's already been translocated. So this will now transfer the nascent chain through the gap in the channel and cause the chain to be disengaged from the channel. So now you'll have a nascent chain no longer associated with the channel. Instead, it will be lodged in the adjacent bilayer. And the C-terminal domain of the nascent chain will continue to be synthesized by the ribosome. The ribosome is oblivious to what's going on. Eventually, coming to a chain-terminating codon, one is left with a protein no longer engaged in the channel, but with an N-terminus in the lumen of the ER, a single membrane anchor domain, and the C-terminus facing the cytoplasm. So this then is the final topology of a type 1 membrane protein. It's now fixed in its orientation, and in that orientation, it will be transported eventually to the cell surface, where the N-terminus, as I've illustrated before, is topologically the outside of the cell. Okay, so this relies then on an event in the middle of biosynthesis that causes the nascent chain to disengage from the channel. Okay, everybody follow that. All right, now suppose I told you that within this type 1 membrane protein, there happens to be a sequence at this position that has the asparagine X-threonine serine characteristic of an N-glycosylation site. Will this type 1 protein be glycosylated? Yes, it will be glycosylated because this part of the molecule has been exposed to the lumen. If this sequence happens to be in the C-terminus, and that's the only place in this protein where that sequence is found, will this protein be glycosylated? No. Okay, now suppose we did an experiment where we took the gene for this protein and deleted the sequence that codes for the membrane anchor domain. What would happen to the protein during its biosynthesis? Yes?
The entire protein would be
right. And if this, in that circumstance, if the C-terminal domain had the sequence asparagine X-threonine or serine, would such a deletion mutant protein be glycosylated or not?
Yes.
Yes. Okay. Right. So again, this sequence is context dependent. It is accessible if it's in the lumen, not if it's not. Okay. So now that's the type 1 membrane protein. Those are very common. Most proteins that we'll consider are of that orientation, but not all of them. Some of them, curiously enough, are type 2, in the type 2 orientation. So let's start this cartoon over again. And a type 2 protein is backwards in the sense that it has its N-terminus facing the cytoplasm and its C-terminal domain facing the lumen. Now, there are two mechanisms for the biosynthesis of a type 2 membrane protein, and I'm just going to go over one of them because it's the simplest to understand and explain. Most type 2 membrane proteins begin their life as a completely cytoplasmic protein with no N-terminal signal peptide. So the ribosome synthesizing this protein, it behaves as though it's completely polar cytoplasmic protein. SRP is not involved. The ribosome may not even be attached to the ER membrane. However, in this class of protein, the ribosome synthesizes a stretch of a sequence that is hydrophobic. So there's no N-terminal signal peptide, but in a common type 2 protein, there may be an internal hydrophobic domain, just as we discussed for type 1, but in this case, without an N-terminal signal peptide. So in this circumstance, the type 2 protein keeps a substantial portion, indeed the entire N-terminal domain, in the cytoplasm, whereas in type 1 that N-terminal domain was in the lumen. And in this situation, when the internal membrane anchor domain is exposed, it attracts SRP. SRP is fooled into thinking this is a signal peptide. In fact, SRP, all it really requires is a small stretch of hydrophobic residue, so it can bind to an internal signal peptide. So this is an internal signal peptide. SRP binds to this as it emerges from the ribosome and draws the nascent chain to the translocation site. So now you have a situation where a substantial N-terminal domain remains on the cytoplasm, and now you have a hydrophobic stretch that sticks into the channel as a loop, just as I described for the membrane anchor domain of a type 1. But now the consequence of this insertion is going to be a little different. As this extends, as the chain gets longer and longer, the loop will expose the hydrophobic portion to the gap between the subunits of the Sec61 channel, and this part of the molecule will, just like a signal peptide, transfer through the gap into the adjacent bilayer. But the next portion of the polypeptide that's just been made and inserted into the channel, in this circumstance, is polar. It's hydrophilic. And so it remains in the polar channel. And just like in the case of a signal peptide, as I've described, this guy now has its hydrophobic segment out of the channel, but the rest of the molecule remains in the channel until the nascent chain is completed in its synthesis, and then it just continues along its merry way, now extending the carboxyl terminus all the way into the interior, into the lumen. So in this simple example of a type 2 membrane protein, one lacks an N-terminal signal peptide. The N-terminus remains on the cytoplasmic face of the membrane, and the C-terminus is translocated through the channel. So nothing up my sleeves. Use a variation on the same signal hypothesis, and you can explain the opposite orientation for a large class, a large class of membrane proteins. Okay, now, imagine we took the gene for a type 2 membrane protein, and we appended to the N-terminus, to the very N-terminus, a signal peptide. What would happen to that protein? What would that protein look like? It would be a type 1 protein. Yeah, you could turn a protein inside out by adding a signal peptide. Likewise, if you took the gene for a type 1 protein and you simply deleted the N-terminal signal peptide, you would turn that protein into a type 2 orientation. So the critical thing to remember here is that a stretch of hydrophobic residues at the very end terminus will dictate the insertion of the succeeding N-terminal domain through the channel, whereas in the absence of such an N-terminal signal, an internal stretch of hydrophobic residues can also engage a nascent chain via SRP to a channel and cause the succeeding, in this case, C-terminal domain of the protein to be translocated. Questions? Perfectly clear. Probably not. Yes?
So when you said that you added a signal sequence at the N-terminus and that inflates it,
Yeah.
doesn't the internal signal sequence then get stuck in the membrane again?
It would. If you add an N-terminal signal peptide to the gene for a type 2 protein, it will flip it, and that's because the membrane anchor domain of that protein now causes the nascent chain to disengage from the channel and leaves the remaining C-terminus on the cytoplasmic face of the membrane. So in both cases, the membrane anchor domain attempts to protrude through the channel, but when it's fully extended, it's so hydrophobic it causes that portion of the chain to transfer out of the channel. Yes? Yeah, okay, that's another important point. So again, let me emphasize, for a secretory protein or a type 1 protein, there's a stretch of amino acids at the junction between a signal peptide and the mature N-terminus that is a substrate binding site for signal peptidase. In a type 2 membrane protein, where this junction between the apolar domain and the C-terminus would be formally equivalent to a signal peptide, typically those amino acids that are recognized by signal peptidase are missing. So a type 2 protein does not typically experience cleavage by signal peptidase so that it remains intact. Yes?
Is it possible for the protein to have like the complex and they have those hydrophobic core in the C-terminus for type 2?
At the very C-terminus.
Yeah, well, around that. Because like I was thinking if they have the hydrophobic core, then as it goes through there, it's going to get shifted.
Yes, yes. So there are some proteins that have a hydrophobic sequence right at the C-terminus. And those proteins actually use a different mechanism for their insertion. They don't employ this channel. There's another channel that's recently been discovered that simply plants the C-terminus right in the cytoplasmic face of the ER. But that's a slightly different mechanism. Okay, so bear this in mind, or sort it out, and then I'm gonna stretch your understanding of this just a bit more, but I won't bother to try to conform to this diagram. I'm going to tell you now about how the rules that I've just given for these two types apply in understanding how a polytopic membrane protein weaves its way in and out of the ER membrane to achieve a successive trans-bilayer orientation. So I won't attempt to draw the channel for this, but let me just depict the sequence of a gene that may encode a polytopic protein. And if you can see these, when you sequence genomes, you find some predicted open reading frames that have multiple hydrophobic lengths. So this would be transmembrane domain number 1, transmembrane domain number 2, and transmembrane domain number 3, with the carboxyl terminus and the amino terminus bordering these three segments. Now, in a typical polytopic membrane protein, there may also be, at the N-terminus, an N-terminal signal sequence with a signal peptidase cleavage site. But the succeeding transmembrane domains will lack such a signal peptidase cleavage site. So the rest of the protein will be synthesized and remain intact. Now, let's go through the type 1 and type 2 models that I've just described, and I'll explain then how these two different principles allow one to predict, just from reading the sequence of a gene encoding such a complex protein, how it may thread into the membrane. So let's take it one length at a time, one segment at a time. If we consider the N-terminal coding region of this gene, the signal peptide will be exposed when the protein is synthesized, and the hydrophilic domain that follows will be synthesized. And where will this part of the molecule be transferred? Will it remain in the cytoplasm, or will it be transferred into the lumen of the ER? Lumen, okay. All right, let's go on then. The protein is not so simple. There's a transmembrane domain. This transmembrane domain will attempt to insert as a loop into the channel, and that will cause the chain to, what will happen to this chain? It'll come out and embed in the membrane such that the next polar portion of the molecule will rest where? Where will it be? Cytoplasm. Okay, so up to this point, this is a type 1 membrane protein. All right, but it gets more complicated because now, instead of terminating at this point, the nascent chain exposes yet another membrane anchor domain. And in this context, what will happen to this second transmembrane domain? What will this do? Up to this point, the chain has become disengaged from the channel. But now this thing comes along, and what does that do? It brings it back to the channel. SRP will re-engage this, which will cause this now to protrude as a loop into the channel. It will re-engage the channel. And as such, when it does that, it will cause this portion to do what? What will happen to the next portion? It'll go into the lumen. And so on. So you can see, depending on the number of transmembrane domains that are displayed in a linear sequence along the length of a polytopic protein, you alternately insert, disengage, insert, disengage, and weave the protein in and out through the Sec61 channel. So this portion of the molecule will be in the lumen. This will be in the cytoplasm. This will be in the lumen. And the last bit will be in the cytoplasm. So you have a kind of a serpentine winding of the protein in and out of the membrane. Okay, so now let's do a thought experiment. Suppose I told you that you had this gene and you made a deletion mutation that removed this segment, and then these two polar domains, this one and this one, were now linked directly in the absence of TM2. With such a truncated molecule, where would the C-terminal amino acids rest? In the lumen, right. What about the N-terminal mature amino acids? Where would they be? In the lumen. Okay, so you would then turn the protein inside out, distal to the deletion event. It's like you're changing the reading frame when you delete a nucleotide and you're reading an open reading frame. You throw off the reading information in the distal segments. In this case, you throw off the translocation reading frame in the portions distal to the, in this case, deletion mutation. Okay, so think about it. I think it's it's a little complicated, but I think it's easy to understand if you remember that the two different contexts in which a signal can present itself. A signal can present itself at the very beginning, at the N-terminus, or it can present itself in the middle. And it can do so repeatedly. And there are some proteins that have as many as a couple of dozen membrane anchor domains in one polypeptide chain. So they weave in and out of the bilayer many times during their biosynthesis. Questions on this? Okay. All right, so that's it for this part of the discussion. I'd like to turn to the subject of vesicular traffic, which is the subject that I would normally use to follow this before we talk about lysosomes. But it all ties together, as you'll see. All right, so now we've only gone through, I know this is a little frightening to think about, but we've only gone through the very first step in the course of a protein that must be secreted outside of the cell. So in the pancreas, when you're making insulin in beta cells of the islet, proinsulin, a precursor form of insulin, is threaded through the Sec61 channel, but it's a long way from the lumen of the ER to the outside of the cell. There are many stations along that pathway that require elaborate machines to capture proteins, as you'll see, into small vesicles and to deliver them between different stations in the cell. Quite an elaborate machine that all eukaryotes have to convey molecules from their site of synthesis through the Golgi apparatus and eventually to the cell surface. Literally 100 or more proteins are involved in different stages along this path. I'm obviously not going to go through all of them. I want to try to convey just a few principles of this, and we'll start with the very next step, which is what happens when a membrane protein or a secretory protein is folded properly in the ER membrane and then must be sorted, some to be secreted, others to be left behind in the ER. So here's a depiction of the ER membrane, and here's for instance, the Sec61 channel. The channel itself, Sec61, is what is referred to as a resident ER protein. That means it's designed to stay put. Obviously, the Sec61 molecule must itself be synthesized. And when it's synthesized, it folds in the ER membrane, and it stays there. It isn't transported anywhere. That's where it functions. On the other hand, membrane proteins, such as a protein like this, which is, in this case, a type 1 membrane protein. We'll call this a membrane cargo protein. Or a soluble secretory protein, completely in the lumen of the ER. These two types of molecule must somehow be segregated and transferred out of the ER into the Golgi apparatus. What is it that permits these proteins to be distinguished from resident proteins like the Sec61 channel? Well, membrane proteins have yet another signal. Not the signal peptide that we've discussed that's involved in translocation, because remember, that's present in the precursor molecule, but then it gets clipped off, so it's not there. Once a signal peptide has been removed, that information is gone. So a protein that needs to leave the ER must have a different signal, another signal. Very often for membrane proteins, these signals, which I'll refer to as an ER exit signal, are at the very end of the protein exposed to the cytoplasm. For a type 1 membrane protein like this, the ER exit signal is very often a sequence of several amino acids near the carboxyl terminus on the cytoplasmic face of the ER. So that's a signal that is necessary and in some circumstances one can show that it's sufficient to cause that protein to be packaged into a vesicle, and I'll tell you in a little bit how that happens. Of course, soluble secretory proteins that are completely within the lumen of the ER, molecules like insulin, completely soluble, sequestered, hidden within the membrane, they may have a signal, but that signal is not going to be available on the cytoplasmic face of the ER membrane. The signal for these proteins must be part of the protein, and that signal must be decoded by a receptor molecule in the ER membrane that itself has an ER exit signal facing the cytoplasmic surface. So membrane proteins expose a signal directly on the cytoplasmic face, and secretory proteins expose a signal that is decoded and displayed indirectly on the cytoplasmic face of the ER. Let me give you an interesting example of such a receptor. There are very few of them that have been discovered, but one interesting receptor was discovered on the basis of a study, a human genetic study, on a rare form of hemophilia. So you know that there are a series of a dozen or so proteins that you have in your blood that are responsible for a cascade of blood clot formation. Two of the proteins, called factors 5 and 8, are glycoproteins that are secreted by the liver and are necessary for blood clotting. It turns out that there's a rare form of hemophilia where both factors 5 and 8 are missing. Now that's kind of odd. Why should a single gene mutation cause two different secretory proteins to go missing? It turns out that the locus responsible for this hemophilia is an ER receptor, just like this, that exposes a binding domain to the lumen of the ER that is selective for factors 5 and 8. And human mutants defective in that receptor produce these blood clotting factors 5 and 8, but they stay in the lumen of the ER and they fail to be secreted, or at least secreted very rapidly. So that's an interesting and fairly selective disease, because this receptor seems not to be generally required for other secretory proteins. For instance, these patients with this rare form of hemophilia are otherwise pretty normal. They secrete insulin, they secrete everything else, they secrete antibody molecules. Almost everything that must employ this pathway gets out quite normally, but they have this very specific deficiency, which implies that there may be many, dozens of different sorting receptors, each one devoted to a cohort of cargo molecules. Okay, so that's just the general principle. There must be a sorting receptor, and that receptor must display something on the cytoplasmic face, just as a membrane protein displays something on the cytoplasmic face. Okay, now, what is the machinery that recognizes the signal? We've talked about a signal, but there obviously, just like in the signal peptide, there must be some machinery that recognizes this distinct signal. And that export machinery is a set of cytoplasmic coat protein molecules, collectively referred to as COP2. COP standing for coat protein. All right, here are the steps that involve the assembly of this coat and its recognition of a signal. In the ER membrane, if we have a membrane protein that's got an ER exit signal or a receptor that's carrying along secretory proteins, this signal is recognized by one of the subunits of the COP2 coat, which in turn is delivered to the ER membrane after an initial activation event shown here. The process begins by the recruitment of a small GTP binding protein called SAR1. It's similar to RAN. It's similar to other small GTP binding proteins, but this one is uniquely devoted to this particular event. It's soluble in the cytoplasm, and in its resting form, in its inactive, dormant form, it's bound to GDP, but it is recruited and binds to the surface, the cytoplasmic surface of the ER membrane, through a transient interaction with a guanine exchange factor. This is the SAR1 GEF. So just like the RAN GEF, the SAR1 GEF, which in this case is a membrane protein, causes SAR1 to discharge GDP and pick up GTP, all in the cytoplasm. And the activated form, SAR1 GTP, sticks to the ER membrane. So that activation event is followed by the successive assembly of two layers of the COP2 coat. An inner layer, which binds directly to SAR1 GTP, and another subunit of the inner layer that binds directly to the ER exit signal. So this inner layer begins to assemble when SAR1 is activated. It binds not only to SAR1, but also to the ER exit signal. And once that is bound, then an outer layer of the coat polymerizes to form a network on the cytoplasmic face of the ER that eventually forms a dimple in the membrane and physically deforms the membrane into a sharply curved vesicle. This polymer, this network of COP2, wraps around the vesicle and pinches the vesicle from the ER membrane, producing a vesicle that now is enriched in cargo molecules and depleted of ER resident proteins like Sec61, which simply lack an ER exit signal. So you physically segregate cargo molecules from resident proteins by virtue of the fact that cargo molecules have a sorting signal that allows them to be captured into a polymerizing network, and the network segregates that cargo and pinches the membrane to form a vesicle. Initially, the vesicle is surrounded by a layer of this coat structure, the COP2 coat. But as the coat finishes the pinching event, GTP is hydrolyzed by SAR1, causing SAR1 GDP, which no longer has an affinity for the membrane, to fall off, and the rest of the coat follows so that the coat is shed from the vesicle. So SAR1 GTP promotes coat assembly. SAR1 GTPase promotes coat disassembly. So one is left then with a vesicle that has an enriched supply of cargo molecules. And of course, you will have, at the same time, in these vesicles, secretory proteins by virtue of the fact that the receptor proteins that captured secretory proteins have their ER exit signals also decoded by the COP2 coat. So now, let's say you do an experiment with cells from a patient that has this rare form of hemophilia. And you make COP2 vesicles from ER membranes from, let's say, the liver of such a patient. Those vesicles that you produce with COP2 will have almost all of the normal cargo molecules, but they will lack the blood clotting factors that were dependent on this sorting receptor for their capture. All right, now let's do a thought experiment. I've given you that the sorting signal on a membrane protein is important for its transport, for the transport of the cargo molecule. Tell me an experiment that you would do to test whether such a signal is sufficient for cargo transport. What's the experiment you would do to test its sufficiency? Same kind of experiment that we had on the quiz, but you'd have to set it up in the right context. What if you took the sorting signal for a COP2 cargo molecule and you stuck it on the C-terminus of a cytoplasmic protein? What would that do? It's a subunit of the COP2 coat.
That recognizes the signal first?
Yes. But it only recognizes it once it's been recruited to the membrane by SAR1 GTP.
So SAR1 comes near it and then...
SAR1, when it binds GTP, sticks to the ER membrane. And that acts like Velcro to draw the inner layer of the COP2 coat to the membrane. And once it's plastered on the membrane, it adheres to the ER exit signal on cargo molecules. Go back to the question. If you have that sorting signal and you put it on a cytoplasmic protein, what would that do for the cytoplasmic protein? Any guesses? Right, so that it can't work because it's not in the proper context. So now back to the original question. How would you test whether such a sorting... Well, let's do the two experiments. How would you test whether the sorting signal was necessary for transport and sufficient for transport of a cargo molecule? You have a type 1 membrane protein. What would you do? Yes? And then what would happen to the mutant protein from which you deleted the signal? So it would stay in the ER. Okay? Now what? How would you test if that signal is sufficient for transport? Yes? Okay, you could do that. Right, everybody hear that? So you could take a cytoplasmic protein, turn it into a type 1 membrane protein by inserting, or a type 2 protein, by inserting a membrane anchor domain in the middle of the gene, and then stitch on the exposed domain that faces the cytoplasm, the ER exit signal, and see if that now causes this very synthetic protein to be transported. Another yes? Yeah, that's an easier way of doing it. So you could, in principle, try to plant the ER exit signal, for instance, on the Sec61 protein, and see if now you cause Sec61 to be unphysiologically transported out of the ER. And in fact, these kinds of experiments have been done, and that's precisely how one defines this kind of a signal, whether it's necessary and sufficient, and what the nature of the signal is. Okay. So now let's go on, because that's only the first step. What is the fate of this vesicle? Where does it go? How does it get where it's going? And what happens to the cargo molecules next? Now we have a cargo molecule, and we encounter, in a typical eukaryotic cell, its first target, which is an early compartment of the Golgi apparatus. We'll discuss the organization of the Golgi apparatus next time. It's organized into 3 compartments, typically: cis, medial, and trans. And the cis compartment is the first station that acquires secretory and membrane proteins from the endoplasmic reticulum. So we've got cargo molecules in this vesicle, and they have ER exit signals facing the cytoplasm. But how does this vesicle know, out of the dozens of different targets in a cell, how does this vesicle know that it's supposed to land and merge by membrane fusion with this cis compartment? Well, now I need to tell you about another kind of a protein, a very important protein, that promotes membrane associations in cells. These are the so-called snare proteins. So this is a vesicle snare, vesicle or V snare. And its corresponding target or T snare. So every vesicle that's delivered to every different destination in the cell must have, as part of its cargo, a special V snare, and its target membrane must have a complementary so-called T snare. And when the vesicle then docks at the surface of the target, these come together in close apposition. Bridging the vesicle and its target membrane very closely, and there's quite an elaborate machinery that I'll tell you about in another couple of weeks, that bridges these membranes very close and causes the two membranes to merge by vesicle or membrane fusion. So these come together, and the two membranes fuse, which brings the cargo molecules now into contact with proteins that are in the cis membrane of the Golgi apparatus. So the snares touch, they cause the membranes to adhere very tightly, and that forces the membranes to fuse with one another very selectively. Yes?
Does that vesicle used to be part of the ER membrane? How is it that it becomes charged with the V-snare?
All right, well, the V-snare, like other cargo molecules, must have an ER exit signal, and that promotes the packaging of the V-snare selectively into a COP2 vesicle. So it's just, in that sense, just like other cargo molecules in that it becomes enriched in that membrane. Okay. Now, what you're left with then, of course, is a Golgi membrane that has the V-snare that's now planted in the cis Golgi and the T-snare together. But what the object now is to get this V-snare out of the cis face of the Golgi. You want to get it back. You want to get it back to the ER so that it can be reused. After all, this molecule, the V-snare, is not itself going on to the cell surface. It's there to direct this vesicle during this early phase of the pathway. It's not going to be used subsequently. There must be other V and T snares that inform other membrane binding and fusion events. There are a whole bunch of them. So this one, the ER exit V snare, must be recycled. It's like an escalator. It goes around and around, and it must be returned. So how does that happen? So now let me tell you about a complementary, in the last few minutes, a complementary vesicle traffic event that serves to retrieve proteins that are designed to function back in the ER. There's another vesicle budding event mediated by a different coat called COP1. and a different GTP binding protein called ARF1, a relative of SAR, but a different one, that is responsible for budding vesicles from the cis face of the Golgi into which the V snares are captured for traffic back to the ER. So these molecules are then recovered and transported back to the ER, and there must be, at this surface, another kind of interaction between a retrieval vesicle, which will have its own unique V snare, and a T snare that is designed to function for the retrieval limb of this process. So you have a V and T snare for forward transport, and a different V and T snare for backward transport, and you must have then distinct forward transport signals and distinct backward transport signals. So I'll review that again, but you can see that each part of this pathway must have its own set of cognate V and T snares.